Yes.
Okay, great. So thank you so much for having me. It's really a privilege to be here. Thanks, Chris, for the introductions. Just to repeat, my name is Turner Baker. I'm a project manager down in Sinai BioDesign and also a PhD candidate in clinical research here at the Icahn School of Medicine. So what I'm going to be talking about today is a mission that's been developed by Sinai BioDesign since its inception several years ago, and that is leveraging academia to enhance and expedite device development. And we're going to be focusing on one device in particular that we're very excited about coming up soon, which is the chill cooling catheter. So before I get too far into it, I wanted to acknowledge a bunch of people on the team who have been able to make this whole process possible. This is probably not fully comprehensive, but everybody here on the screen has been extremely important. Dr. Anthony Costa, who is the previous director and founding director of Sinai BioDesign, really brought me into the team and made this whole project possible with our group. And wanted to thank Drs. Christopher Kellner and Hazem Shoira, who were the original surgeons who brought this technology and clinical need to Sinai BioDesign and have been really implicated on really every step of the way of developing the device, bringing it into animals, and we'll talk about all that coming up. Dr. Ben Rappaport, who's the new acting director of Sinai BioDesign and has been an awesome collaborator and champion of this project since joining and working with us. The department has been obviously integral in the development of this whole device. Dr. Bedison for making Sinai BioDesign possible and all the projects that come out of here. And then we wanted to give a special thanks to Dr. Jay Mako and Dr. Dan Gajach, who have been really heavily involved in making sure this project is successful in what's needed in the field. And a special thanks to Dr. Mako, who helped put together these slides and organize everything. So I won't go through each of the individuals down below, but needless to say, the whole project would not have been possible without a group effort, especially my fellow members of Sinai BioDesign, who have helped with really every step of the process. So to dive into it, the process of moving a medical device from what's just an idea or a need in the operating room to actually getting approved, being commercially successful, and coming back into the operating room is a long process. I'm sure everyone's heard from tech or anybody that's done any kind of innovation about the valley of death, which is what occurs in between the basic research of a new device and eventual commercial success. And so the way that we think about it a lot down in Sinai BioDesign is sending out rockets, right? And so the technology is like the thrusters or the engine that really get you off the launch pad. But you have a long way to go before you actually get to commercial success. And there's a lot of things that can go wrong. And if you don't plan ahead, there's this whole period where you're just kind of in the void. And if you haven't generated the resources you need to be successful, you're going to struggle through this period and you're going to rely on luck. And so our goal at Sinai BioDesign is to minimize that. And so even though a lot of our technologies, the thrusters are very good, a lot of things can go wrong. There can be failure to actually launch the device successfully, but even those that exit successfully from an institution or an incubator, they have lots of problems, right? And so there needs to be some life support systems built in that can help these projects be successful and navigate to where they're trying to go consistently and with as much ease as possible. So what do we mean when we talk about life support systems for medical device development? So what we're talking about is leveraging the resources that are available within Mount Sinai and the Icahn School of Medicine to de-risk the device itself and to make it more attractive to outside investors and outside entities. And so we do this through a various number of mechanisms, including developing the intellectual property to make sure the white space that we're growing into is available and protectable, doing foundational research that establishes that the science is sound as well as the technology itself is feasible, prototyping, which is perhaps what Sinai BioDesign is best known for, doing rapid iterative prototyping, doing benchtop testing to test out those prototypes and make sure that they're feasible and functional, and integrating surgeon feedback heavily into the whole process so that what we're growing into is an actual need and not just an engineer's daydream. and then testing those devices in animals, which are available to us. And eventually, once we have a later stage prototype, generating key opinion leader support from both inside and outside. And all of this is really available at our fingertips within the Mount Sinai health system. So what BioDesign tries to do is leverage these resources and this availability, oftentimes in the same building, to make this device development pathway quicker, more cost effective, and overall just enhanced. So our idea is that instead of just being a simple rocket launch, we're really trying to build the spaceships of the future, right? That will make this path towards commercial success consistent and easy, and we know what's going to be going on there. So I know I've been kind of speaking in dorky rocket analogies as well as some vague terms. So what I think would be best is we're going to go through a new device that we've been developing for a while that we think will be spinning out into a startup in the coming weeks and months, which is the chill cooling catheter. So the chill cooling catheter is used in conjunction with minimally invasive ICH evacuation. So this is where, as I'm sure many of you know, after a hemorrhagic stroke, the clot is accessed via minimally invasive techniques and is aspirated out. Unfortunately, at the time of the initial injury, a secondary path of cell death and edema begins to occur, and that occurs even after the blood has been evacuated. So the idea is to develop a combination therapy that can tackle the secondary injury and can be deployed during the surgery, during the evacuation. And so with that in mind, we developed the chill cooling catheter. This is a closed loop fluid-based catheter that induces cooling from within the cavity that's left over after the hematoma has been evacuated. And so the future of the treatment, we really believe, is going to be that you evacuate the hematoma, that you deploy the chill catheter, and then you chill just locally around the area of the brain. And so the first thing that we did as we began to develop our life support system to help this project be successful was ensure that there was intellectual property available and that there was white space to grow into. And so this underwent kind of a classic biodesign approach that we use internally, which is that the project was brought to us by Drs. Hazem Shoira and Christopher Kellner, who had proposed an infusion cooling catheter to be used similar to what has been seen in some ischemic and cardiac spaces. This went through the biodesign process, which eventually led to a more technical description of the device, as well as developing broad methodological claims. And eventually we looped in Mount Sinai innovation partners who we worked very closely with and filed for provisional patents, both at the US Patent Office, as well as the PCT cooperation, the Patent Cooperation Treaty, which is the international patent system. And so now feeling confident that we had intellectual property, kind of like our launch pad that we know we can jump off from, we undertook some foundational research to make sure that the science is sound as well as the technology. So this began by doing a systematic review of both preclinical and clinical studies that had been conducted looking at therapeutic hypothermia to treat ICH. So we did a meta-analysis of the preclinical studies, which had local and systemic cooling, so where the body is either cooled in entirety or just the brain is cooled. And what we found is that there was an overall impact of reduction in edema and improved neurobehavioral scores in response to the therapeutic hypothermia, really across the board. We also saw that there was not a significant increase in hematoma volume, which is good because a major concern is coagulopathy issues. And so looking at the clinical results, again, we see significant edema reduction across many of these studies. However, there are very high complication rates, including over 96% of pneumonia in the cooled patients in some of these cases. And we really feel like this is because of the systemic nature of these studies, and that ICH patients can't undergo systemic cooling as successfully as, say, a cardiac patient where there has been overwhelming success with therapeutic hypothermia. So having done this, we really felt confident that our technology was needed and that it would likely be successful and efficacious. Having established that, we wanted to look into whether or not the device would actually be functionally feasible. And so to do that, we developed a computational model. It was a finite element methods model. We did this in COMSOL in collaboration with CUNY and some of their heads of their engineering program that have done this before for deep brain electrodes. And so what we did is we developed a model with classic bioheat equations that were informed also from past literature, more recent past literature. And then we engaged in a collaborative work with Monteris Medical. I'm sure many of you in the audience already know about them, but what they do is a neuroablation device with MR lit, where it's a laser ablation device, and then they track heat in real time using MRI. And what they do before they turn the laser on, though, is they activate a cooling probe. And since they track it in real time, when the laser is not activated right away, it gives us a unique period of time where we can look at intracranial cooling, similar to how the chill cooling catheter would work. So what we did is we took this data and we compared it to the model that we had built. And making a few adjustments into our model, we were able to successfully prove that the proposed bioheat equation FEM model closely matched clinical data for intracranial cooling in the same space using the same type of probe. And so then we also looked at this model in terms of what's clinically relevant, which is breaking out the area of cooling that you see around the probe based off of mild hypothermia, so below 36 degrees, moderate hypothermia below 34 degrees, and profound hypothermia below 32 degrees. These are relevant as mild hypothermia is anything under 36, so you know that you're thermomodulating that area, that tissue. Profound hypothermia is important as that's where there are concerns around coagulopathy, and that's especially important in being used in ICH, where rebleeds are a serious concern. So having established the model and validated against clinical data, we now wanted to look at our device and say, what do we need to do in terms of our distal end probe design, and what do we need to design so that we can achieve the thermomodulation that we want to see? And so we did that and looked at the volume of tissue cooled, so you can kind of see where there's the region of profound cooling versus the region of mild cooling. And we went through several different iterative processes to figure out what exactly we needed. And some of these tests are ongoing to figure out what kind of temperature of influx we need. But overall, it's been great, and it really led to a lot of the developments of our final current design. So having established that the science is pretty sound, that we're trying to grow into, and that the device that we've come up with and now patented is likely going to be functional and is designed to be optimized, it was time to start prototyping. So this again is really what BioDesign is known for, and it occurs over an iterative process from some very rough in the lab works to something a little more formalized. And then after testing, we eventually reached an MVP. And you can see here that we now have an inflatable balloon catheter with a highly conductive force, albeit a bit maybe wiggly in the video. We have a detachable lure mechanism. And then we have a thermopump that's powered by a Peltier device to induce the controlled cooling. So now, having established everything prior and with a prototype in mind, we wanted to engage in benchtop testing to make sure that this actually worked in the real world. So to do this, we developed an advanced phantom model down in BioDesign for ICH. So here we made a blood substitute and put it into a previously developed hydrogel brain that we have downstairs. You can see that we were able to inject around a 30 mil hematoma into the brains and put that into a validated school model that we've used previously. And so what this allowed us to do, you can see here on the left that there is some CT images of the pre-evacuated hematoma, as well as the evacuated hematoma with the device in place. We're able to just in our lab conduct these surgeries and practice all of the elements that we're talking about for what the device is going to be used for. And this is really key downstairs as it helps us recognize problems such as some of our earlier designs had a permanently affixed lure mechanism. This obviously cannot be tunneled. And so we learned this very quickly, and we're able to develop new detachable, attachable lure mechanisms, which ended up being very important for some of the provisional patent literature. But eventually, we hit a wall where the engineers can only do so much, and we need to go upstairs and engage in the experts, the surgeons themselves, to inform the device and make sure that what we're going into is a real medical need. And so we did just that, where we brought the phantom model and our current device up into the operating rooms and angio suites and engaged with the experts. So here you can see Dr. Kellner using the phantom model. On the right, you can see this is the hematoma being evacuated. This is our model that we built in-house. You can see that our device is able to be deployed through standard surgiscopes that are used during current ICH surgeries. And then the surgiscope can be removed so that the catheter can be left in place. The device can then be tunneled. The brain is able to be cooled in the hydrogel phantom model. We can do controlled rewarming, and then we're able to remove the device similar to how you do with EVDs, so without another surgery. And so over time, and with some changes iteratively to the prototypes as we went along, we're able to confirm with Chris that each of the steps involved with developing this device, and every step that's going to be used in the future procedure, is achievable by our current prototype. So knowing that it's now functional and can be used in the OR, we wanted to actually go put it into an animal model. And so that's what we did. We got a sheep model to try this out in. And this is really where the whole pipeline of developing it all in one place and the benefits of being inside of an academic situation like this are so beneficial. Because in a single building here in Annenberg, we have everything that we need to develop devices. Down in the MC level in the basement where BioDesign is, we can do the prototyping and develop the phantom models that we were discussing. We can just go right upstairs to the 8th floor and engage with the surgeons to leverage their expertise and get feedback on the device. And then as a unit, we can just go right upstairs to the 26th floor where they're fully equipped to do animal studies, large animal studies with experts in animal trials and with a lot of imaging equipment as well. And so all of this together means that there's a lot of iterative design that can happen and synergistic efforts that can lead to better results. And so this is really why we feel that we can launch a lot of great projects out of here, de-risked with animal data attached. So for the chill device, we were able to induce an ICH into the sheep brain using autologous blood injection. We let edema form for about 3 hours before conducting an evacuation using the same tools that are used in human surgeries. We're able to deploy the catheter through, again, the surgiscopes that are also used in human surgeries, remove the surgiscope around the device, and then attach the detachable lure mechanisms to induce the bidirectional fluid flow. We were able to cool the brain down for 3 hours. You can see our MVP down there cooling the brain. And then we tracked cooling for a bit in real time using MR thermometry, which is what Monteris uses during their MR lit procedures. And then we were able to remove the brain for later histological analysis. So the model was able to successfully mirror edema formation within the brain, and the device also was able to cool successfully. So you can see down here, this is the MR thermography data, where the blue represents the radiated cooling induced by the cooling catheter placed within the cavity. You can also see that it extends beyond the area of edema that we were marked. And so here you can see that data tracked out, similar to how you saw it with the computational model. The y-axis is the area cooled around the probe, so it's a 2D over time. And so you can see here that the Monteris cooling probe, which they use to combat the laser ablation, was tracked with some error bars. And then our N of 1 sheep study with the chill device outperformed that, showing that it was better at cooling pretty rapidly right away at the site of injury. So now, having established that the device is functional, it's workable in the OR, it can be used in a living system successfully without safety implications, but with proof of feasibility and efficacy, we wanted to engage in key opinion leaders, both inside and outside the both inside and outside of Mount Sinai. And so the reason for this is that obviously we want people to be excited about the device, but we really also want to understand how this will be integrated into human trials and into the clinical setting. So questions like how long should we be cooling for? What's the optimal temperature? and which ICH patients are most likely to benefit from this type of treatment were all the questions that we still wanted to know. And so we wanted to engage with experts who had done therapeutic hypothermia in the space of ICH. And so what we did is we formed the Hypothermia for ICH study group, or the HITCH study group. This was led with Dr. Kellner, myself, as well as Dr. Stefan Mayer. Dr. Kellner obviously has a deep history in ICH evacuation. Dr. Stefan Mayer has led and published a number of protocols and research into therapeutic hypothermia across a broad range of strokes, including ICH. So what we did is we reached out to other experts that have published a lot in the space and are known for it, to ask them, what's the best strategy for doing intercavitary cooling as opposed to other types of cooling mechanisms? Which patients should we be recruiting into a first-in-human study? And what is their general opinion of the chill device, especially now when we can still do some prototyping. We haven't reached design freeze yet. And so this is the group that we put together. I won't go through each of these individually, but everybody here, you might recognize some of these names, has provided some foundational research into therapeutic hypothermia, and especially in the space of ICH. And so it was a great group with dynamic discussions. And the first meeting was actually on Christmas Eve, so you can tell that people were excited about it. And so this is actually the first slide that we opened with during the study group to get people in the right mindset of chilling. So the meeting occurred, we had 6 meetings over 3 months with this group. And again, we broke it up into 3 main phases that we wanted to accomplish. So this included general introductions and kind of having an open forum discussion for people to talk about what they wanted in the space, as well as introducing the chill device and getting some feedback. We had a leadership group that led discussions for each of the 3 phases. So this first one was led by the original organizers, Dr. Kellner, Dr. Mayer, and myself. The second phase was to figure out what's the best cooling protocol. So how quickly should we cool people down? When should we cool people down? And what's the target temperature that we should be aiming for? We asked to help lead the group discussion 2 members of the team, which were Dr. Fred Colbert and Dr. Kees Polderman. Dr. Colbert is really the foundational preclinical expert and has done numerous mouse studies, rat studies, looking at cooling them down to treat ICH. Dr. Kees Polderman was really foundational in establishing therapeutic hypothermia in the space of cardiac, post-cardiac neuroprotection. And so these guys were great for leading this meeting. The final question that we wanted to answer again was, what's the best trial strategy, and what's the best controls, what's the best outcome, who should we be recruiting? And so for this was led by Dr. Rainer Komar and Dr. Fred Rincon. Both of these 2 physicians have led clinical trials before. You're looking at systemic hypothermia in human ICH patients, Dr. Komar in Germany and Dr. Rincon here in the States. And so they were both great resources to lead the discussion on how we should think about an intercavitary cooling protocol, trial protocol. The results were great from the discussion with the full group. And we had general positive feedback of focal cooling following evacuation with one of the groups. I haven't gotten his approval yet to put his name on this, but it was certainly said that they said that this is the true path forward for using therapeutic hypothermia in ICH. And so that was very exciting and reassuring. During the second phase, we helped nail down what we believe is going to be our future recommended protocol for our first-in-human studies, which is that we are going to do a combination of systemic normothermia and localized hypothermia, and shooting for a target temperature of 33.5 to avoid any early issues of coagulopathy, though there was some hearty discussion that there might be benefits and it might be very feasible and still safe to do even lower temperatures than that. It was recommended that at minimum we want to do around 48 hours of cooling, with early cooling being the most important, which fits in very well with some of the other research going on here at Mount Sinai, like the MINUTE trial that was recently submitted, and the idea that we should be rapidly evacuating these patients. To look at the best trial design itself, a lot of great stuff came out of this, and we actually got everyone to discuss a rollout approach for what the trials would look like, including a feasibility trial with no control arm, a pilot trial looking at local versus systemic cooling, and then a more pivotal trial where we're actually going to start to show the efficacy that we want for some of the labeling of our device, such as reduction in edema progression and improvement in terms of neurological decline and functional improvement. So overall, the meeting was really great, and everyone was engaged and excited about the product. And to take a step back again and think about, as we launch products out of Sinai BioDesign and Mount Sinai in general, we're excited to continue to do stuff like this and lead study groups where hopefully the idea is that these members will be excited about the technology and serve in some capacity as a scientific advisory board or similar for CHIL. And so now that the CHIL package is fully put together, we're really excited about it and feel like it's ready to launch out into the world and be successful as it tries to make its way towards commercialization success. Looking forward for CHIL and for certainly other products coming out of BioDesign, we want to add a few more value adds to this package. This includes doing first-in-human trial protocol design. So we already kind of initiated this with the study group, but we want to formalize this and come together with a fully written protocol so that everything leaving the institution as a startup has a written and vetted protocol from Mount Sinai, which is a huge value add. We also want to start engaging earlier on with FDA interactions and getting direct feedback from them from pre-submissions. This is especially because the FDA is explicit about the fact that they are more lenient with academic institutions in terms of early discussions and setting guidelines than they are with industry partners. So there's a huge benefit in doing this from within an academic institution. And as they like to say, you should go early and often so that you know which milestones you need to hit. And so CHIL is obviously a very exciting technology that we're looking forward to see enter into the real world. But this concept of building these packages, these life support systems, we're going to be applying to really all of the products that are coming out of Sinai BioDesign in the coming years. We think that this is really just going to set Mount Sinai up as the successful launch pad, the Cape Canaveral of medical device launches, especially in neurosurgery, as we move forward. The other additional benefit that I just wanted to close on, in addition to obviously successful medical device startups and products, is that a lot of the stuff that we did to research and bolster this project are other benefits that have been added to Mount Sinai. And so the brain model that I discussed has actually become its own product that was successfully sold to groups like Integra for conducting training leading up to their own clinical trials in ICH evacuation, and other ICH devices. When testing our tunneling mechanism, we learned a lot about the fact that EVDs and similar catheters slip out of the brain in neurocritical care situations. This has initiated a BioDesign device that's being co-led by Drs. Kellner and Dr. Dan Gajec. And so that will hopefully get its own packaging and testing before it exits successfully. And the work that we've already engaged and started to do with our first-in-human trial protocol for the CHIL device has undergone a lot of work and actually gained attention from the NIH's Trial Innovation Network. And so there will be, there's a publication out with our collaborators at Johns Hopkins, Tufts, and Harvard that will be coming out soon, outlining the way that we're hoping to roll CHIL out. So I just wanted to close again with some acknowledgements and thank everybody on the team, especially the BioDesign group, my fellow basement dwellers, for helping lead this whole project to the success it's already had and success we hope to keep having. So with that, I wanted to thank you again for letting me present and ask if there was any questions.